this part 1 workshop today, and my name is Joanna Porankiewicz-Asplund. I've worked with Agrisera for over 27 years, and I have been focused on antibodies, Western blot, troubleshooting, and antibody validation. And I will present to you examples of what can go wrong in Western blot technique, how to fix it, and you are very welcome to ask me questions. You can write them in the chat, or you can write them at the end of this workshop. So there will be question and answers session. There will be also a quiz. The prize is going to be a freely chosen antibody. If you are a winner, you can choose an antibody from our catalog. So let's begin. First, a short introduction. Part 1 is today, and part 2, I remind you, is next Thursday at the same time. A little bit about Agrisera, which is located, as maybe some of you know, in northern Sweden. So the winter day looks like this, with hopefully beautiful northern light. And in a summer night, we have a constant light. So there is a very short and intensive season for plants, but absolutely amazing. On a map, you can see where we are located. And it is Agrisera has been around for quite a while. We celebrated 40 years last year, and we are having over 80 employees and 2 facilities. One antibody production facility outside Umeå, where we are producing polyclonal antibodies, and one facility in Umeå from where our antibodies are shipped all around the world. On the pictures, you can see my colleagues from the lab, and we are also ISO certified, which is important because all documentation is conducted with the ISO standards. Agrisera, as you know, has been around for a while. Agrisera antibody collection has been was created 25 years ago. And we were awarded a Best Supplier Succeeding in Plant Science award last year. As you may guess, our products are in thousands of publications. So we provide antibodies to a variety of plant proteins, hormones, epitope tag antibodies, secondary antibodies with various labels, and we are cited in over 10,000 publications. So on our product information sheets, you will see a long list of references for quite many of our antibodies. And what is our agenda today? I will cover the following steps of the Western blot. So sample extraction, protein sample collection, extraction, gel electrophoresis, and we reach until the transfer. And next week, I am going to cover the remaining steps, including also interpretation of Western blot results, which sometimes can be a bit difficult. So that technique, which is called Western blot, is a visualization and measuring of specific protein in a complex mix. This is what we're doing using Western blot. I wonder if you know how old this technique is, because it actually has been already first published in 1979. So the technique has been around for quite a while and is still extensively used in laboratories all around the world. But why does it have a name Western blot? You're probably familiar with Southern blot. So this is about DNA work. Then we have Northern blot for RNA. And then we have Eastern blot for post-translational modifications. And Western blot for proteins. As you can see, it is a little bit like in the rows of winds on a compass. And the name Western blot comes from the laboratory which first worked with this technique, which was located on the West Bank. So therefore, the Western blot. And the principle of this technique, just to remind you, we have a so-called transfer stack where we are applying the current. First, we are electrophoretically separating proteins in a gel, polyacrylamide gel, and then applying the current and transferring them to the membrane, and analyzing with specific antibodies. You will receive also PDF of this presentation with links to various resources. Protein transfer is exactly this, what you see in this picture. What we are after is that we are getting a mirrored copy of what we had on a gel. Actually, that is often overlooked, as we are thinking that maybe the antibody is bad, the extraction didn't work, but actually the protein transfer may fail and may be a reason that you're not obtaining good results with your antibodies. Remember, we need to get a mirrored copy of what we had on a gel. And I need to remind you a little bit about antibody structure, because this is going to be very important later to understand how antibodies are actually detecting specific proteins in the Western blot. So the antibody is a protein which is a Y-shaped molecule. This part, so-called Fc region, is called like that because it's a fragment which crystallized. When this IgG molecule was at first researched upon, it was left in a fridge and it crystallized, and therefore we have a name Fc region that is non-changeable region also, which facilitates antibody purification using protein A or protein G. However, what is the most interesting and important is this part. It's the antigen binding domain. That's a variable part of the antibody, which is going to fit to a certain sequence on your antigen, in this case, protein. And the mammalian antibodies are built in a similar way. They look exactly like that, like a Y-shaped molecule with the most important region, which is this antigen binding domain. And that region will also influence the stability of the whole antibody. Therefore, some antibodies may last for years. We had antibodies which were maintaining their activity for over 40 years, but some antibodies may actually lose reactivity and deteriorate with time. Let's look closer at this antibody binding domain, which is located here. And this is a very important thing to remember for the future, that we have two types of epitopes. So antibodies may bind so-called linear epitopes, which, as you see here, are subsequent amino acids in protein linear structure. And an epitope can be anything from 2 to 15 amino acids. And these epitopes antibodies are binding to in Western blot in immunohistochemistry, where proteins are denatured. However, antibodies are also recognizing so-called conformational epitopes. So these continuous epitopes, so we have distant amino acids which are brought together in a 3D fold of the protein. And these epitopes antibodies are binding to in techniques like immunoprecipitation, Western blot in native conditions, and CHIP. So please remember, antibodies bind to epitopes which can be linear or conformational. And this is crucial for later work with the antibodies. And antibodies are poly or monoclonal. There is a myth saying that monoclonal antibodies are always better, but that's not exactly the right thing to say. Polyclonal antibodies, poly means many. So these antibodies are going to bind to multiple epitopes. So polyclonal antibodies can be produced in those different species, and polyclonal antibody is a pool of several epitopes. So antibody can bind to stretches of amino acids of 2 to 15 amino acids in the linear structure of a protein, or they can detect the conformational epitopes too. Monoclonal antibody will bind to only one epitope. Mono means one. And this has an influence later, because if monoclonal antibody is losing activity, then you will not see any signal at all, because there is only one epitope which is detected. With polyclonal antibodies, we can see gradual deterioration because they recognize a pool of different epitopes. So remember, epitope is 3 to 15 amino acids. We have a polyclonal antibody, which is a pool of several epitopes. They are very versatile and robust reagents, and much cheaper to produce compared to monoclonal antibodies. And when you're choosing antibody for your work, please consider first at the desk if you have access to the right antibody. This is very often overlooked. Antibody works for Arabidopsis, and it is assumed it's going to work for other species. And this is not the case. I will show you why. We can predict both reactivity to a protein in a certain species, as well as antibody reactivity in specific application. So you can save your precious time in the lab if you first determine if you are using correct antibody. This is absolutely crucial and will save you a lot of work and resources. And how to do it? So, example, we have a photosynthetic protein, PSBA. In green, you have marked the parts. So we have an N-terminal part, we have C-terminal, and the sequence to which antibody was made is chosen here on N-terminus after signal peptide. We have a sequence chosen from inside of the protein, inside the fold, and then we have a C-terminal sequence. If that sequence is not found in the protein you are aiming to detect, the antibody is not going to work. And as you see, it can be determined without doing experiments. It is also very easy to obtain false positive results with the antibodies. So before you use any antibody in the future, please check if the sequence it is made to is found in a protein you're aiming to detect. If it is an antibody you received from a colleague, you need to ask what this antibody is made against. Very often, a full-length sequence is blasted and aligned, and based on that, it is assumed that the antibody is going to work. This is not correct. And this data may not be an available exact sequence on the antibody product info sheet, but you are always welcome to ask a supplier to provide you not maybe the sequence, because that's covered by intellectual property, but you can always send the protein sequence you aim to detect, and a supplier can tell you if the antibody has so-called predicted reactivity. As you may guess, you can also predict if the antibody is going to work for native technique, because if the peptide is chosen from not exposed region, the antibody is not suitable for CHIP or immunoprecipitation. So you can save a lot of time when you analyze the sequence the antibody is made to. And here are small reminders in those boxes for you in the future when you receive the presentation. So remember that antibody can have both predicted reactivity to a protein from a given species, as well as we can already, based on the sequence, we can determine if antibody is suitable for technique, which is native, or a technique in which protein is denatured, like immunohistochemistry or denatured Western blot. When is the antibody suitable for multi-species detection? Because that sometimes we work in a lab with many different species. We use Arabidopsis thaliana, maybe rice, Chlamydomonas, and it would be nice if one and the same antibody work for the same protein in those different species. And we designed so-called global antibodies, which are made to a very conserved region, in this case, PSBA again, and that peptide is repeated in protein sequence, PSBA protein sequence from those different species. So with one antibody, you can detect target protein in a wide range of species. So you're saving resources, and then you can hopefully obtain also comparable results. Of course, the level of the detection will be varied between the species depending upon physiological state of the sample. Here you can find a link to a quantitative Western blot video tutorial, which can help you if you would like to use antibodies, global antibodies for quantification work. Some of our global antibodies are accompanied by protein standards, and then quantitative Western blot is a suitable technique. I often get this question: will knowing the antigen sequence help to identify cross-reacting bands? And as you understand from what I said, the sequence is unique to the protein. However, there may be coming unwanted cross-reactions which are rather connected to the experimental setup to the Western blot protocol. And from the sequence, which is unique when we are choosing the peptides, they are absolutely unique for a given target protein. We cannot identify the cross-reacting bands. Only mass spec analysis or sequencing can tell you what protein is in that band, although you need to remember that in Western blot, in the gel electrophoresis, in one direction, SDS page 1D, there will be hundreds of proteins in each band. So absolutely not, we cannot determine which cross-reacting protein we can see on the blot, knowing the sequence the antibody is made to. If you ever attempt to order a custom antibody in the future, because of course we have so many model species and each is carrying thousands of proteins, and suppliers are offering antibodies to a small percent of targets. But therefore, if you would like to make a custom antibody, I would like to stress that it's very important to do so-called pre-immune serum screening. So you need to ask the supplier to provide you with this service. I will show you here why is it so important. So each sample is number 1, Arabidopsis thaliana, number 2 is barley. And then we have used serum from non-immunized animals on this blot. And then you can see that some animals already have antibodies to certain plant proteins. Of course, as you may guess here, at 50 kilodaltons, we're seeing Rubisco. So if you're, for example, attempting to make an antibody to protein, which is around 40 kilodaltons, you already have this animal is already responding to some protein at that size. Therefore, it's not suitable. So this screening will allow us to choose animals with least background at the time of immunization with the antigen we would like to obtain antibodies to. This is how we do it at Agrisera. And as you know, rabbits are having plant-based diet. So of course, presence of strong cross-reaction with Rubisco is not strange. So please keep this in mind and ask the company which is conducting antibody production service for you to provide you pre-immune serum for conducting this screen and choosing the rabbits with least background. And now we are jumping into the Western blot and antibodies. So we have 2 different options of antibody binding. Primary antibody is directly labeled with enzyme or fluorophore. This method is less common and as the sandwich method. So in the sandwich method, we have target, primary antibody, and secondary antibody, which is labeled with enzyme or fluorophore. And as you see here, we have for one primary antibody, there will be few secondary antibodies binding, and this will enhance the signal. Therefore, this method is much more popular. In option 1, we would require directly conjugated primary antibodies. And this can be done in the lab very easily. There are kits for that, but the option 1 is suitable for proteins of moderate and high expression. If your protein is of low abundance, this is the first choice to use. And this is also the most common technique. And now, Western blot flow. Well, it's many steps. Many things can go wrong. But the outcome can be also different. Following this link, you will have step-by-step protocol with different explanations. I am focusing here not on technicalities, but I would like to show you how to solve the issues when things go wrong in Western blot. So this complicated procedure with many steps. And what is the outcome? As you see here, maybe some of those blots you recognize yourself, you have seen it in your own lab. There was the same antibody, primary antibody, used in all 4 laboratories. The analyzed sample is Arabidopsis thaliana, but you see the outcome is very different, right? So one lab gets no signal at all, then background, a lot of background. Finally, one laboratory is detecting target protein. Target protein was quite challenging because it was transcription factor, and transcription factors are not so easy to visualize using antibodies, and oftentimes nuclear fraction needs to be analyzed. And I will show you on a specific example how to do it. Why? So how much do you know about the protein you aim to detect? You need to start with this. Before you do any Western blot, you need to ask yourself this question. How much do you know about the protein you are aiming to detect? This is your checklist. Biochemical properties, isoelectric point. Because if you use the buffer at the isoelectric point of a protein, the protein has no charge, right? Expression level. Is the protein highly expressed or is expression level very low? In which organ or tissue is this protein localized, and in which compartment in the cell? But you may ask me, how do I know about the protein level I am analyzing? Is it even possible? Yes, there is a very useful database for that, which I'm using daily. It's called Protein Abundance Database and HACS. And here you can check not only for Arabidopsis thaliana, but also for other model species. You will be able to check what are the levels of the protein in various organs. This is based on experimental data, and I highly recommend to use this database because over the last 40 years, we have learned that if a protein abundance is below 10 ppm, it is going to be quite challenging to detect such protein with an antibody. And you may think that you have bad antibody, but actually the problem may be the protein abundance. Sample quality is crucial here. We know now the abundance level of our protein, and now we need to extract it. And we can use various model species, plants, algae, or cyanobacteria, and our protein during extraction was going to be denatured. Following this link, you will find references to various extraction protocols. And also, if you need free images, we have free images of model species for download, just as a present for you. So we have high quality requirement for protein samples, and we have different extraction methods. We have time efficient extraction method, which is a bead beater, often used for nucleic acid work. It can also be used for proteins. We here in this blot, we compared bead beater versus sonication, and it was basically no changes in protein level depending upon this which technique has been applied here. We have more time-consuming extraction method as mortar and pestle or sonication. Sonication is connected with heat generation, and it's not my favorite extraction method. Mortar and pestle is efficient. However, if you have 20 samples to extract, your hand is going to get tired. So efficiency of extraction is not going to be comparable between samples. It was reported by one of researchers who obtained results which were very promising in his 15 samples, but then he swapped the order of analysis. So sample which was extracted last in the first experiment was extracted as the first in his next extraction round. And it appeared that observed pattern was due to extraction, not to biological difference between these specific samples. So please be very careful when you're choosing extraction method, and you need to work, of course, in the cold, and in some cases, also proteins can be sensitive for light. So for example, extraction can be done only with a dim green light. Some proteins may require specific inhibitors. I will mention it later. So you need to prepare theoretically very well before you go to the lab and conduct protein extraction. We have prepared this poster, which you can find on our website, which is focused on exactly protein extraction and is leading you to pages with specific methods which are dependent upon the organs, also water content, and so on. So on protein extraction, one could make another separate seminar. And for proteins of low abundance, let's say you went to PAX, you checked your protein, your target protein is around 10 ppm. It means that you may need to do fractionation. And doing cellular fractionation is time consuming. There are kits for that, but they have varied efficiency. However, many antibodies are discarded because they are only tested on total cell extracts. As you can see in this example, this is a photosynthetic protein, and when specific fractions have been used, the target is concentrated. So this is a photosystem 1 protein, and when fractionation has been done to isolate photosystem 1, the target band has been visualized. Please remember, for proteins of low expression, it will be very difficult to obtain signal in total cell extract. Well, you may ask me what to do if a protein is cytoplasmic. Well, in this case, I would recommend TCA acetone precipitation and very high protein load, between 100 to 300 microgram per well. So there are always some solutions, and if you are not certain what to do, you can always ask me. I hope I convinced you for fractionation. And protein extraction methods may involve a choice of specific buffer, of a correct buffer. As you see in this example, first the test lab got no band at all. By adjusting the buffer, most commonly buffer which is used contains tris, is tris-based, but for this protein, HIPIS was working much better. And here we have an example where the peptide is conserved in all those 3 species, barley, Arabidopsis, and rice. But in the first blot, we have only band for Arabidopsis. What is the difference? 6 molar urea has been added to extraction and loading buffers. And what this reagent is doing is that it is denaturating the protein, unfolding it further, and making it accessible for the antibodies to bind. Some laboratories are, as default, always using urea. Of course, it has also negative sides because it can be connected with smearing, and there can be other issues, but it can always be optimized. So buffer composition is extremely important. You need to use specific organ. For example, pin proteins, pin 1, pin 2, will not be detected if a very tip of a root is analyzed. We need to include protease inhibitors, but also proteasome inhibitors. There was a lab which was for 1 year trying to visualize a protein until they realized that it is targeted by proteasome. Once proteasome inhibitor was included, the band was visualized. Of course, we need to optimize our protein extraction to get good yield and use sometimes specific conditions, like almost dark. For example, I would like to stress here that protein extraction has to be done very promptly because protein pattern may change during extraction, and the more steps are added, the worse it may become. So protein extraction is your key to success, and the antibody may not always be to blame. And how are you working? Good laboratory practice is very important when you work with Western blot. What does it mean? We are having it clinically clean, cold, and precise. When did you calibrate your pipettes last time? It is very important to fulfill those 3 criteria. I got asked if there is any difference between which gel we are using, because there are different gels, precast gels offered by different suppliers. And there may be a difference, as you can see here, that you may get better transfer, for example, for certain proteins, depending which gel you are using. Agrisera is using Novex gels, which give a possibility to modulate the gel electrophoresis depending upon which buffer it used with one and the same gel. So this can be different between different precast gels. Sometimes it may look like this. So there is an advantage of using precast gels. Of course, they are more expensive. However, they will give you a high chance of reproducibility of obtained results, because precast gels are produced in controlled conditions. What do we load per well? Yes, I'm very curious to know what you're loading. Are you loading based on protein or on volume? So recommended, absolutely recommended, is microgram of protein or microgram of chlorophyll for photosynthetic organisms. Not recommended is volume. This is, I'm amazed how often researchers don't know how much protein is loaded per well. And if a protein is of low abundance, this will be quite important to know how much of a protein in microgram has been loaded to a well, because then we need to load more. As you heard, it may be required to load 100 to 300 micrograms. And I often hear that, but how do I measure protein concentration in the presence of components of extraction buffer? And there are reagents which can help you to do it. We are using this BCA protein assay reagent. There are other reagents available, but they are compatible with detergents, with the naturing reagents, with SDS. So that way we know what we are loading per well, and we can compare different samples with each other. I got also a question if we should load nanograms or micrograms. It also depends what you're loading. If you're loading a recombinant protein, then of course you should use nanogram. If you use a total cell extract, then it is micrograms. And when you establish that the antibody is recognizing correct target protein, then you can do titration. As you see here, the titration, there are two different protein loads per well, and then combined with different dilutions of primary antibodies. So this is the way to go once you know that your antibody is working, and then you can find the best optimal dilution for obtaining a clean result. And the gels can be 1D or 2D, and how does it affect the results? It depends what you are after. What questions are you asking? 1D gels are most often used for Western blot because we are separating proteins which are charged with SDS based on their molecular weight and then transferring them to a membrane. But as I mentioned, you need to remember each such band will contain hundreds of different proteins. Well, 2D gels, we first separate the protein based on molecular weight, and then we are placing that little gel piece, and then we are separating all proteins in the band, which will then provide us with a spot pattern. So depending what you're asking, you may use either 1D or 2D gels. There are differences between those approaches, and it's of course possible to make Western blots also on the 2D gels. And now sample denaturation. There are different possibilities here. And as you may guess, we are using 70 degrees for 5 minutes. Boiling the samples extensively in these high temperatures is a very standard procedure. However, some proteins may aggregate. So if you're unlucky, your target protein, for example, H plus ATPase, may aggregate when you heat it at 100 degrees, which means that it will be stuck at the top of a gel and never enter, therefore no signal with the antibody. Protein transfer conditions are also important to consider, and I will provide you with a quick guide what to do. As you see here with Western blot, you have to prepare a lot at your desk before you go to the lab. Quite often it is not done, which results in quite a lot of replications of the blot after blot after blot, while the work should be done theoretically. So please keep your blots always in the cold. If you don't have a cold room, you can run it as we do in the ice bucket with cold buffers. And then protein conditions are transfer conditions are dependent upon molecular weight of your target protein. So here is a quick guide for you. Look here, if protein is above 20 kilodaltons, the pore size of a membrane should be slightly higher. Also, higher molecular weight protein prefer less methanol, but more SDS. And wet tank transfer is more preferable for proteins of higher molecular weight. So this is a quick guide for you to set up correct transfer conditions. The main mistake is to use the same conditions all over again for each protein. But proteins are like people, they are different. So when we are doing the transfer, which membrane are you using? There are two choices, of course. We have PVDF or nitrocellulose. And most laboratories are using PVDF, which is more robust. Both membranes can be air-dried, but PVDF needs to be rewetted and activated. Nitrocellulose is more fragile. Actually, which membrane to use depends upon your protein-antibody pair. I have seen results for some proteins and some antibodies. Results were better with nitrocellulose. So ideally, one should have both membrane types in the lab. And if something doesn't work, then change to another membrane type and see if there will be an improvement. Membrane activation with methanol is the first choice. However, there is a concern about toxicity of the methanol. Therefore, you can actually replace, and this is what we have done. We have replaced methanol to ethanol. But you should also, when you're doing it, you should compare if you have already set up protocol and you know what detection pattern you obtain, then you should compare those two. Protein transfer type, of course, we have two possibilities, wet or semi-dry, and there are differences between them. So when it comes to amount of buffer, of course, we need to have more for wet transfer. Please remember not to reuse any buffers. There's another electrode contact, and we have another speed. And I cannot tell you which one is better because both work. However, wet transfer is less prone to failure. So if you are a beginner, you should start with wet transfer. Semi-dry transfer requires a little bit more skill, and there can be both working very well for your protein antibody pairs. It has to be simply experimentally proven. And protein transfer, when we are doing it, we should of course, remove the air bubbles from when we are building the sandwich, but I recommend not to do it like this. I recommend you to set up a sandwich in the cassette. And once you have done it, then remove gently air bubbles. If the air bubble is at the top, for example, we are letting it be. The air bubble will obscure the current and therefore may contribute to difficulty with the transfer only when it is at the range, molecular weight range of your target protein. And here in this video, you can see how we are removing the air bubbles. Protein transfer needs a quality control, and it can be very easily done using those two stains, Ponceau or Air 250. Why to do this control? This is the reply why. Because if your protein pattern looks like this, after you have stained your membrane reversibly, after taking it from the sandwich, after protein transfer, if the pattern looks like this, we don't continue with this membrane because it is very clear here that there was some issue with the sample. The protein pattern should be like this. And this quality control is very easily done. The stains are reversible. You can destain easily with water, and then you save yourself the work. Because if your protein pattern is looking like this, I would recommend not to continue, but go back, review the protocol, and make a new transfer with fresh samples. And there's another control, which is very easy to do, which is very often overlooked. Do you stain your gels after the transfer with Kumasi? Because those gels should show no bands to prove that the transfer was efficient. So as you see here, we have made a double sandwich. So we had two gels in a sandwich and two membranes. And then we stained gel number 1 with Kumasi and gel number 2. And as you see here, there were bands still in a gel number 2, which means that the transfer was not efficient. So if you do this check after the protein transfer, you will know if the transfer was efficiently made and the proteins left the gel. If you get bands in the molecular weight range of your target protein, it means that the transfer was not efficient and you may not have enough of the protein on your membrane to visualize, to detect it later with the antibody. So please keep it in mind. And there's also another possibility here, with low molecular weight proteins, if transfer is too harsh, voltage is too high, it has happened that protein is blasted through the membrane. It will not stick to the membrane. I have been troubleshooting low molecular weight proteins which were blasted through the membrane, and therefore there was no signal when developing the membrane with the antibody. So remember, high molecular weight proteins require different conditions compared to low molecular weight proteins. And this is very often overlooked, unfortunately. And now we come to the antibody format. What you see here is lyophilized antibody from Agrisera, but they can be either antigen affinity purified, or you can also deal with antibodies which are provided as a total immunoglobulin fraction. And please be observant, because sometimes it looks like you're buying a lot, because it will be written 1 or 2 milligram on product info sheet for primary antibodies. But if it is antibody purified to total immunoglobulin fraction, it will be only a percentage of it, which will be specific antibody. In serum, I often get questions how much of specific antibody is in serum. It is not possible to say until the serum is purified and it can be calculated. Total immunoglobulin fraction contains from 0.5 to 5% of this total fraction will be your specific antibody. While when we are dealing with antigen purified antibodies, most of what you have in a tube will be a specific antibody. And just to remind you how the purification is done, this is how we are doing it at Agrisera. We have resin, we are binding the antigen, the protein or the peptide to the resin, and then we're passing the serum through. Specific antibodies are binding, and they are eluted, most often by low pH, which is changing the conformation of a protein or peptide, and therefore the specific antibody is released. And primary antibodies are often reused. They are expensive reagents, and I understand that you want to reuse them. So how many times are you reusing your antibodies? Is it 1 time? Is it 20 times? Actually, there are no golden standards for it, and it depends very much upon specific antibody. So if you are reusing the antibody, primary antibody, you can observe that the signal is getting weaker, because the antibody will bind in each incubation, and sometimes all of the primary antibody will bind in the first incubation, and there will be nothing left for the subsequent one. So please remember that not all antibodies can be reused, and if you don't have a signal in a second experiment, it means that all antibody bound to your target in the first Western blot you conducted. But I know also that many of Agrisera antibodies are successfully reused, as you see here, up to 20 times. But this is something which you have to empirically test yourself. And primary antibody can sometimes lose activity. And it is happening, it differs between the antibodies. It can be different reasons for it. So first of all, storage temperature, something went wrong, and an antibody was defrosted. It is also depending upon purity grade. Actually, antibodies stored in serum format are most stable, and you should never purify all serum, because once you purify, pull out specific antibody from serum, as I showed you, the antibody may be less stable. There can be a need of stabilizers. Also, if you use the same primary antibody multiple times, it may lose activity. So you should always follow the recommendation from a supplier and check carefully product information sheet, and do not add glycerol to serum, because that's not necessary. Serum is the best format for antibody storage. And here we have prepared another poster. If you would like to receive it, you can go on our website. This poster is built based on my presentations, and you will find a lot of QR codes where you can be directed to specific pages. And now, a surprise, we offer 100 free antibodies if they match your experiments, the questions you're trying to answer. Then you are free to check our list and let us know which antibodies are of your interest. There is no limit on provided antibodies, and the agreement is that we received the results, and also we include secondary antibody, high titer secondary antibody, together with the primary. And now there is a time to share with you the link to the quiz, which I would like to invite you to participate in. You can do it later when you have time. The winner will be chosen and announced next week, so you don't need to stress. I will also send the information in the chat. And I would like to thank you very much for your attention. And now there is a time for questions. If you wonder, those pictures are from surroundings of Agrisera. We still have snow up in the north. So welcome for questions.
Yes, so I have a question.
Yes?
So if there is a situation, I'm doing electrophoresis for proteins, and suddenly the power goes off, like electricity is gone. What do I do? I just continue the electrophoresis when the power is back, or do I redo it from the beginning?
Oh, it depends for how long the electricity was gone. Because if it is just a short moment, then yes, you could continue. But if it is, because proteins will be diffusing also. If it is just a short power failure, then maybe like 10, 20 minutes max, I would continue. But if it is a longer one, you need to redo the gel electrophoresis.
But what if I put it not... oh no, I don't know how to say it. Like, what if I put it horizontally, so it doesn't migrate?
Aha, you mean so, but it is not based on how the gel is placed. I think it's based on the physics that it will be simply diffusion of the proteins from, because suddenly the current is gone, so nothing is pushing them through the gel.
Oh.
Do you understand? Do you understand what I mean? So I would redo the experiment.
Okay, I understand. Thank you.
You're most welcome. Here I see the question. I would like to know, what are the most important things to consider when you're trying to detect a protein with EFP, GFP tag from a transgenic plant leaf? Well, the most important is that you have enough of expression, because I think in many cases the epitope tag antibodies are failing because it's not enough of the protein to detect. Then, of course, this is actually a very good question, because it is common knowledge that one and the same epitope tag antibody may fail to detect another tagged protein. And in such case, I would recommend to use urea, for example, to unfold the protein and make it more accessible. However, the key here is amount of a protein you're aiming to detect. So the principle is the same also for working with detection of proteins which are tagged. And also, of course, the whole procedure. So depending what is a molecular weight of your tagged protein, you will need to follow the guidelines. So if your epitope tag protein is 80, 100 kilodalton, you need to apply conditions suitable for gel electrophoresis and transfer for this molecular weight. So you need to basically follow the guidelines which were presented in the frame of this workshop. You will receive the presentation. Are there any more questions?
Hi, Johanna. Thank you first for the workshop. And the question is the next one. In some laboratories, when someone is running the yield, they put it on ice, but it's better to put it in ice or just without it?
Or always cold.
Always.
I mean, yes, always cold. Because if you don't apply cold buffers, and you can just use the ice bucket if you don't want to be in a cold room, you will get the so famous smiley effect. So always cold. Remember, it's better to do it always in the cold. You will get better results. I would not say that sharper bands, but definitely in the cold.
Perfect, thanks.
You're very welcome.
Hi, Joanna. Can you hear me?
Yes, absolutely. Go ahead.
Hi, I'm from the University of Verona with the Bassi group, and
welcome.
we are working on mostly photosynthetic proteins. So I was just wondering if there's any protocol that can allow us to run the membrane development overnight instead of doing room temperature as we usually do, and also about transferring procedure. We usually do 1 hour at low temperature. Is there any possibility that we can run it overnight, maybe at low voltage?
Yes, there is such possibility, but please remember that you have to modulate it. If your protein is of low molecular weight and you run it for too long, it may run out of a gel. So with those controls which I showed you, like staining the gel after after gel electrophoresis and transfer, you can estimate what transfer conditions are suitable. It's always molecular weight of a protein which is deciding what conditions you should apply. And developing the reaction, I will talk about it next week, but I can tell you right now, and I will show you also the examples, that for some antibodies, incubation, primary antibody incubated overnight will lead to background. So for some antibodies, you can do it. Some antibodies will require incubation overnight. I will show it to you next week. But for some, not. So if you are obtaining a lot of background, you maybe incubate for too long.
Okay, thank you very much.
Yes, you're welcome. You can also, if there are questions which I haven't answered today, you are always welcome to send it to me by email. And I see here a few questions more. I would like to ask why, when I stain my PVDF membrane with Ponceau, I see only very faint bands instead of clear signals. I don't know what is the percentage of Ponceau. Maybe you should recheck the protocol. Are you using fresh solution, or has it been around in the lab for a while? Because I think with Ponceau you need to prepare it freshly. And faint bands instead of, so that's what I would be troubleshooting. I have a question about designing the antibody. Are there any difference for efficiency between antibodies raised against peptides or raised against full recombinant protein? Oh, I love that question. There are differences because, as you saw, it is a difference in what antibody is binding to. If you use a short sequence, then the epitope pool will be smaller, right? If you use full-length recombinant protein, you will get more epitopes, but instead maybe more background. Okay? You follow? Because the peptide is unique. And also, I would like to stress here that there are often situations when antibodies rise to recombinant proteins are not recognizing endogenous proteins in the tissue. So recombinant protein approach is not always the most optimal. And if you would like to read about it a little bit more, we actually have been describing it on our website, and I can share a link with you. So, of course, if a protein has a lot of, if it is a big gene family with a lot of isoforms, making an antibody to recombinant protein and the sequence being present in those other isoforms would make a reagent which would be recognizing not a unique protein. So here it is a link to our antibody production guide. And here I have a question about studying Andean microalgae. Oh, exciting. Under different light intensities at the photosynthetic level, if it turns out to be a new species, how can I determine the correct protein sequence? This is a very good question. Well, actually, I would be very careful to use antibodies on a species where we cannot check the conservation of the peptide or protein which was used to elicit the given antibody. So please don't use antibodies on samples where you do not know protein sequence to check if the given antibody can work, because I have seen false positive results with antibodies, even published. It was a publication on some cyanobacteria where Agrisera antibody was showing beautiful band of correct molecular weight. The problem was that this sequence the antibody was made to was not found in that protein. So I would not advise you to use any antibodies unless you know the sequence. But you can do also, you can try to do the data mining, and you can send me, you can connect with me, and I can help you to determine if an antibody can be used or not. My email address is here in the chat. If you would like to send me further questions, there will be a second part next week on Thursday, and I warmly welcome you to join. And thank you for your time today, and thank you for all the questions. It was really great that you asked so many questions. And please take part in the quiz. And I hope to see you in 1 week. Thank you very much for your attention.